Thank you, Evelyn, and thanks so much to Martin and the organizers for having me here. It's so wonderful to be back in Dublin, as you can see from my name, my ancestral homeland here of Ireland. And I know that some of you knew us as Underdog for the first couple of years of our company's life, and we've recently changed that to Cyclarity Therapeutics. So that's just a name change, no big shakeup with the company or the mission, just maturing and moving forward there. It's so intimidating to follow somebody like Dr. Khan and hear him talk in this visionary way about how we're going to transform the world, and then I have to think about, like, I'm going to be like, well, look at this graph, look at the line, it's going up, isn't that cool? This experiment's working, but at the same time, also, it's not just humbling, but it's also important, I think, that we pick apart the problem. Like, aging isn't a problem, it is 1,000 problems. And so we need 1,000 little companies or 100,000 companies and labs going at this one piece at a time. So I'm just going to tell you about our little piece of the puzzle. And we're targeting oxidized cholesterol that accumulates with age. And this is a little summary of a review article that we published a couple years ago about how oxidized cholesterol, the main species of which that forms non-enzymatically by reacting with oxygen-free radicals, is 7-ketocholesterol. And I'll use oxidized cholesterol and 7-KC sort of interchangeably when I talk here. It's a basic. What I want to emphasize is that this is a basic aging molecule that accumulates in various cells and tissues with age. I'm going to focus mostly on the macrophage and atherosclerosis as an indication, and I think we're lucky that we have an indication that we can focus on and that we think will have a big impact in when we go into clinic soon. But since it's a basic aging molecule, it's implicated in Alzheimer's disease, in macular degeneration. We got a grant from the NIA to test our drug in Alzheimer's disease. So when you work on aging and when you target aging, you have many diseases to go after. Just to hone in on this a little bit, to try to simplify and make it not so confusing. You've heard of OXLDL maybe, oxidized LDL. The oxidized cholesterol is a piece of the very complex and heterogeneous molecule or complex that is OXLDL, but it is not the same as OXLDL. OXLDL has a lot of things and a lot of oxidized things. Oxidized cholesterol, this particular species, 7-KC, you don't need it for anything. If I could wave a magic wand and make it disappear from every cell in all of your bodies, you'd just be better off. You wouldn't miss it. It's broadly toxic. It intercalates into membranes and damages the integrity of those membranes. It's a potent lysosomal toxin. And interestingly, it's, and I don't think I'm showing any of the data here, but it's in the blood, inside of the blood cells, not necessarily in the serum. It's very predictive of heart failure. And like I said, it's implicated in most diseases of aging. I'm going to talk mostly, like I said, today about atherosclerosis, depending on which study you read. Atherosclerosis doesn't kill anybody, but it actually kills a third to half of everybody by causing a huge percentage, an overwhelming percentage of all heart attacks, of strokes, of COPD. And that's why when you risk adjust these diseases for atherosclerosis, you're talking about a third to a half of everybody on Earth, not just in the Western world, people eating Big Macs or something like that. You're not free from this if you if you eat a healthy diet and exercise. Everybody builds atherosclerotic plaque with age. Everybody's building up oxidized cholesterol with age. So just one damage molecule that's accumulating with you every day of your life. Our model for how atherosclerosis proceeds is that you have lipids that start forming in the walls of your artery. Macrophages, monocytes get recruited, they differentiate in macrophages. Macrophages, their job is to eat, and they try to eat up the damaged lesion, in this case lipids, and cell debris that are there. However, when they consume too much of it, especially oxidized cholesterol, things that they can't digest, they actually become part of the problem instead of the solution. They keep eating, their lysosomes shut down, and they turn into foam cells, which is a key part of the plaque. And our goal when we set out was to find a way to reverse that. Can we turn the macrophages back into healthy functioning firemen who put out fires instead of starting them? And so we went about trying to develop drugs that could pull the oxidized cholesterol out and make them functional again. So we started out with started out with cyclodextrins. Going back to the literature in the 80s and 90s, you find people looking at these molecules, these cyclic carbohydrates, and their ability to interact with different molecules. So cyclodextrins, a lot of them are really good at being very soluble and sticking and encapsulating targets that are very hydrophobic. So cholesterol and a lot of cholesterol derivatives are very hydrophobic, and indeed you can find cyclodextrins that can interact with cholesterol and different cholesterols very well. So we learned a lot about that. We did a lot of modeling. We did a lot of computational modeling, and we eventually put together that actually having two cyclodextrins in a certain configuration around our target, this oxidized cholesterol form, was the most stable form, and then we engineered a new family of cyclodextrins, these cyclodextrin dimers. I think that it's a good time, it's an opportune time to be bringing cyclodextrins as drugs, as APIs. They've been used in medicine for the last 20 years as excipients, and so a lot of the medicines that you already take have cyclodextrins in them already. Regulators have become very comfortable with them. They can be made to be extremely safe, and so we learned from that. We engineered our drug to have the same safety properties as the most safe cyclodextrins that are already being delivered to people, but we also engineered them to have high specificity and affinity for the target, which traditional cyclodextrins tend to be very weak, and I'll tell you more about that in a minute. So this is a little bit of a confusing graph that we published last year, but we did this. This is a physical screen, an in vitro screen that we did of every cyclodextrin that we could get our hands on, and a bunch of new cyclodextrin dimers that we created. And the important thing is that as you go up here, you decrease the affinity for the target, and as you go to the right, you increase the specificity for the target. Okay, so for our purposes, stuff in this quadrant is the worst, and stuff in this quadrant is the best. So we came up with a bunch of hits for cyclodextrin dimers that had both high affinity and high specificity for the target, at least according to this simple screen, just a simple automated turbidity assay. Here's a summary of a lot of data. Once we picked a lead candidate to start developing, or a pre-lead candidate, we started testing it in a variety of cell types that try to mimic the disease. So the monocytes and macrophages that we're talking about. In the first graph there on the left, we show our ability to pull the oxidized cholesterol out of macrophages that we culture in the lab and load up with oxidized cholesterol. So we can turn them into foam cells by loading them up, and then we can pull the oxidized cholesterol out. Now, the most exciting thing on this slide, though, is when we tested it on plaque tissue from humans. So from patients who had plaque that was removed, we soak it in a solution with our drug, and we can pull it out in a concentration-dependent way. But what was crazy about this is we first did it for like a whole day and for hours, and we noticed that the kinetics were the same. The lines just overlap. And so then we did it for 15 minutes, and it's the same. It comes to equilibrium within 15 minutes. It acts very fast, which is important because the pharmacokinetics of our cyclodextrin, as with others, is very short. And it's also very cytoprotective. So this oxysterol is extremely toxic to any cell type that it comes into contact with, and we can protect. These are monocytes, which are the precursors to macrophages. We can protect them at very low concentrations in a dose-dependent manner. But what about actual foam cells? So I said that we can create foam cells in the lab. We can load them up with lipids like others have done before. We can do it much more potently and quickly by treating them with the oxidized cholesterol. We can do it with ox-LDL. We can do it with naked 7-KC and different mixtures. This is just a summary of one version of this. We can do it 6 different ways. We can prevent foam cell formation. We can do it simultaneously with the insult, or we can actually reverse it. We can turn them into foam cells. So these are foam cells here. These blotches here are like the definition of the foamy lipid droplets that accumulate in the macrophages. And then we can make them go away and turn back into something that looks like a normal macrophage. And that's, I think that's more or less unprecedented. This is all unpublished stuff I'm showing now. Not only that, we can turn them back into functional macrophages. So like I said before, the job of macrophage is to eat. When you turn them into foam cells, that goes down, and then we can bring it back in a dose-dependent manner. So it looks like we're reversing the foam cell phenotype. I think there's a lot of interest in cyclodextrins therapeutically. There's been a couple of publications in the last 5 years, more than a couple, but a couple of big ones that stand out, using very large doses in mice to try to treat dyslipidemia disease. This paper from the Latz group, where they had knockout mice, the cholesterol transport knockout mice, they get atherosclerosis. Normal mice don't get atherosclerosis. That's why they're kind of bad models for it. But when they treat with harrowing doses of this drug, they can reverse atherosclerosis. Now, it happens that there are children with a rare genetic disease that are in clinical trials getting this form. I won't really call it a drug. It's actually on the inactive pharmaceutical ingredient list. And so they're getting enough of a dose that to get the kind of dose that these mice were getting, you'd need a 6-hour infusion to get it, and it might make you deaf. But it might cure your atherosclerosis. And then similarly, another group here at the University of Arizona have a very nice model of stroke, I think nicer than the models of atherosclerosis. And they don't try to prevent stroke. They don't try to interrupt it within the first few hours like the only treatments available for stroke do now. They actually are treating in the days and weeks after the stroke happens and treating the damage that the stroke is as if it's a dyslipidemia disease. Really interesting story. I encourage you to look those up. I don't really have time to do it justice. So we invented our drug is 1,000 times more potent than those versions. So we want our drug to be to actually be a viable drug candidate that you can deliver in reasonable doses to patients in a reasonable amount of time. So this is a summary of a lot of safety data, and this is kind of a summary of all the work that we did so far in the cell-based assays. If you extrapolate from the cell-based assays into the concentrations that you would use in vivo in a human, then this is the dosing range you would be in. This is a log2 scale, so this is a long way over here, and here's when we can start seeing side effects in the mice. So we think we have a large, wide window for delivering our drug, a large therapeutic range over which we'll be able to deliver a safe concentration of our drug to people. So it sounds like I've been talking mostly about basic research so far, but we're actually really far along the IMPD, IND enabling path. And we've had many regulatory meetings already. We've had 4 regulatory meetings so far. in the UK and in the US with the FDA. We've been awarded the ILAP award, which is an accelerated regulatory path in the UK, and so we're leaning into that and focusing for now our clinical development in the UK, and with the close interaction with the MHRA, which is the UK version of the FDA or of the EMA, depending on which one you're most familiar with. We're hoping to bring our first clinical trial to people middle of next year. This is a rough design for our phase 1 trial. Not that exciting, mostly a safety test, and hopefully we don't make anybody sick. We may be able to look at some middle-aged subjects who have undiagnosed pre-disease plaque. So that could give us some interesting data out of that. And then really exciting will be the phase 2 study when we actually get to test patients, and we're going to use next-generation non-invasive imaging to measure plaque as an endpoint in that study. So we're really excited about that. So overall summary of where we've come and where we're going. We launched at the end of 2019, and we've raised $14 million to date. We're going to need another $30-ish million to run those 2 clinical trials that I just described to you. So we'll need to raise that money later this year. This isn't a solicitation for funds, but if you are involved with investment networks that might be interested in that, come talk to me. We're doing manufacturing. We're already at kilogram scale manufacturing level. We're about to start our GMP manufacturing that will go into people next year. We've already done a complete set of non-GLP safety testing. We don't want any surprises. And now we're doing the GLP safety testing in animals. And we've had all these regulatory meetings that I already mentioned to you, and of course we have more in the future. And the next big hurdle for us will be applying for IMPD probably early next year. And we're funded well within being able to apply and hopefully receive IMPD next year. So I've been talking about just one drug, and honestly, that's most of what we have and the overwhelming data that we have. We think that our drug, like I said before, oxidized cholesterol, it accumulates in lots of cells and tissues with age. So we're working a little bit on Alzheimer's disease. We're interested in stroke. We're interested in this rare orphan disease that's a cholesterol transport disease that hyperaccumulates oxidized cholesterol. We're interested in macular degeneration, and we're interested in what else our platform can do. Can we design new cyclodextrins that can bind other small toxic hydrophobic biomolecules? Can we design a cyclodextrin that can bind A2E and other oxysterols, for example? So those are some of the things that we have in development. We have this iterative platform that goes back and forth between the computational and the bench lab, rapid screening process. Since I have a few minutes left, I'll try to talk about our computational platform. Now, bear in mind that my level of computer programming stops at maybe being able to write a loop function in BASIC. So fortunately, I have geniuses who work for me who came up with this, and Mia Anderson in particular, who is the head of our computational chemistry program as an intern in 2017, she proposed the idea of, hey, I took a class on modeling things in school. Why aren't you modeling these? And I said, what do you need to try this out? And she said, I need $100 for a software license. And I said, give it a shot. And here we are 5 years later, and she's working on her PhD simultaneous to being a co-founder of our company. She can do large-scale screening of different pairs, so host-guest interactions, target and cyclodextrin, whole matrices of these in large-scale, fast, cheap screening methods. We can do the higher resolution molecular dynamic simulations, and we've designed this pyramid that we're using to screen for new cyclodextrins that can target anything that's small and hydrophobic, but we're only really interested in small, hydrophobic, toxic molecules that accumulate with age. And so we're finishing optimizing this and implementing this platform as we speak, and we're getting it all the way to the point where we can predict affinity constants in silico for a given cyclodextrin for a given target before we even synthesize a single molecule and test it in the lab. So I'll stop there. I'll thank the group, some of our advisors, our funders, and especially all the people who do the great work. And I'll take questions if there are any from the audience. Thank you.